Hey guys, today I'm going to be telling you about the cell cycle, how it happens, and the differences between mitosis and meiosis. So what is the cell cycle? Cell cycle is a process that most of our cells go through, not all cells, most of our cells go through in order to duplicate themselves. These are the 5 phases of the cell cycle. We're going to start at G0, the rest phase. Some of our cells don't replicate at all. They'll stay at G0 throughout our entire lives, but this is when the cell is not actually replicating. It's simply just doing its normal procedure, its normal job. However, whenever the cell gets a signal in order to start replication, we'll go into the G1 phase. This is what's going to start promoting cell growth, and the cell cytoplasm, the cytoplasm organelles, are going to start being replicated through the G1 phase. We then go into the S phase, which is DNA replication, in order to split that between the cell we are going to split into, followed by the G2 phase, preparing for the final phase, mitosis, which is the actual splitting, and lastly, the M phase, which is mitosis or meiosis. The M phase is a very, very small portion of the cell cycle, but it is very complex and is actually split up into 5 different phases that we will be talking about later. So, you also see these 3 little checkerboard flags. These are checkpoints throughout this overall cell cycle in which the body is just going to make sure that the cell is going through the cell cycle properly so that we don't get a poorly made cell that's constantly replicated. So we have, first off, a G1/S phase checkpoint. This is just going to make sure that the organelles from G1 were made properly, aren't going to be duplicated improperly throughout a new cell. We've got a G2 checkpoint, which is just making sure G1S and G2 all went properly, especially the DNA replication part. And then we have a third checkpoint during our M phase, during metaphase, which I'll tell you about later, just making sure that all the chromosomes are going to be aligned properly. So, what do these checkpoints actually mean biologically? How do we tell that a cell is going through this process properly? Well, we can do that through cyclin. More specifically, CDK, cyclin-dependent kinase. Here's a small drawing of what CDK may look like in our bodies, and our cell has all of these CDK. They are currently inactive, which means they're not going to be doing anything. The way to tell if a cell has gotten through a checkpoint is how much activated CDK is in that cell. So if a cell is going through the proper cell cycle, it's going to have an increased amount of active CDK up until the point where it passes the checkpoint. It's activated through cyclin. Go figure, cyclin-dependent kinase. And cyclin is what's added into the cell as it's being told that it's going through the checkpoints properly. So here we have our little drawings of cyclin, and it's activating our CDK into what's known as MPF, a maturation promoting factor. All right, now we're going to talk about this M phase, because this is the most complicated phase of the cell cycle. And we're going to talk about the 5 phases that it's split up into, starting with prophase. Okay, so what happens during prophase? During prophase, the nuclear membrane dissolves, the centrioles divide into opposite ends of the cell, and the chromosomes become visible. They become tangible in the shape that we know them to be. So here in our drawing, you have the typical cell, and you can see this is the cell nucleus. It's starting to dissolve. It's starting to just go away so that we can have access to this genetic information over here. Here we have the chromosomes, the 2 chromatids that are attached to each other, as we know, sister chromosomes, and the 2 centrioles on opposite ends of the cell. These are going to be very important for the M phase. Next up, we have metaphase. So what happens during metaphase? The chromosomes are going to align in the center of the cell. They're also going to be attached to spindle fibers created by the 2 centrioles on opposite ends of the cell. So here's our drawing, and here's what's going to be happening. Our 2 centrioles on opposite ends are going to be attached to these spindle fibers. And the spindle fibers are going to attach to the chromosomes, straight down the middle. And you see the chromosomes are all aligned perfectly just like this. And what's going to end up happening is that the two sister chromatids are going to be detached and separate to opposite ends of the cell. This is where we also have that third checkpoint. We want to make sure that all of these are aligned properly before anything else happens. If it's not aligned properly, the cell is going to not split the way we want it to. Some cells aren't going to have enough chromosomes, which is very, very, very bad. In mitosis, which is the process being described here, we're going to have the 46 chromosomes being split. However, it's still going to be 46 and 46, just chromatids, haploid cells. Anaphase. This is the actual splitting of the chromosomes here. So, we're going to talk about how this happens. Chromosomes are separated into chromatids and moved to opposite ends of the cell, as you can see in the drawing here. We have the 46 chromosomes being separated into two 46 chromatid segments. You'll see the spindle, they're being separated across the spindle fibers. So when we actually look at a chromosome, this is what it looks like. It's the two sister chromatids, and they're being attached by something called a centrosome. This is how we're going to separate this, because we're going to break down this centrosome. Our body has a lot of something called separase. Go figure, it wants to separate the chromatids. It wants to dissolve this centrosome. That's its purpose. However, all of the separase in our body is currently inhibited by something called securin. Go figure, it wants to keep the chromatids secure. It wants to keep this together. So it's going to inhibit separase into this little compound that I have drawn here. So this is what's mainly floating around in our bodies. It's a separase inhibited by a securin. When we actually want to separate this chromosome, our body is going to produce something called APC, which is anaphase promoting complex. It's this little bomb that's going to go around and it's going to flag the securin. And the body is going to look at the flag securin and it's going to destroy it. So it's going to be flagged and say, destroy me. So we're going to go through this process backwards. So the securin is flagged. The securin is going to be destroyed off of the separase, and we're going to get separase, which is going to dissolve the centrosome. And once the centrosome is dissolved, this is what we're going to end up with. This is how anaphase is done. Up next, we have telophase. Telophase, proper organelles are moved to the opposite ends of the cell, such as mitochondria, stuff like that. It is the last step before cytokinesis. It's the last step before the cells are actually going to be physically separated from one another. And this is when we have a formation of either a cell plate or a cleavage furrow. Cleavage furrow is in animal cells. Cell plate is in plant cells. So here are our two new cells. You can see the single chromatids in each one, followed by the nuclear envelope starting to develop and the other proper organelles in the cytoplasm. And then we have this little separation. This is our cleavage furrow / cell plate. This example, it's going to be a cleavage furrow, and it's made out of mitotic spindle fibers. So this is going to be what's separating the two cells from one another before we actually split up in the process known as cytokinesis. This is the full separation of two genetically identical cells. And this is what we're going to end up with. We're going to end up with two cells that are in the G0 phase. Just simply doing what their purpose is supposed to be done, whether it's like a liver cell, a heart cell, you name it. All right, one more thing. This entire process is known as mitosis. Most of our cells go through the process of mitosis. They split up 46 chromosomes into two cells that each have 46 chromosomes. Each of these are still 46 chromosomes. It hasn't actually lost any. We're going to talk about meiosis, which is different from mitosis. It actually follows the exact same rules as we had before, except it's got a few more steps, and we're going to talk about that. So meiosis is the duplication of specifically sex cells. Instead of creating two daughter cells, two genetically identical cells, it's going to create 4 genetically identical cells, each that have half the number of chromosomes as the original. So the original had 46. We're going to end up, after meiosis, with 4 daughter cells that have 23 each. Okay, so For meiosis, we're going to have prophase, metaphase, anaphase, and telophase. And we're going to have each of these as prophase, metaphase, anaphase, telophase 1. And then we're simply just going to duplicate that a second time in prophase, metaphase, anaphase, and telophase 2. The only real thing that's different during meiosis is metaphase 1 and metaphase 2. So, if you'll remember, we have metaphase during mitosis. This is what the chromosomes are going to look like. We're going to have all 43 that are aligned just like this, and we're going to split these chromosomes in half. The difference in meiosis during metaphase 1, the first metaphase, is that the chromosomes are actually going to look like this. We're going to get one chromosome on top of another chromosome. And this is where crossover can occur. It can actually cross genetic material. So once this splits, we're going to split right in half. And so this is where we're going to go from 46 total chromosomes to 23 and 23. 23 and 23 diploid chromosomes. Finally, we're going to go into metaphase 2, which is the same as mitosis metaphase. It's the same chromosomes are aligned like this. We're just going to have 2 of these cells that duplicate into 4 total daughter cells, each that have 23 chromosomes, half the number that we started with. Remember, this is specifically in sex cells. So, remember the rule. Mitosis made mitos, and meiosis made me.